Hi everyone, and thank you for taking the time to listen to my presentation today, which is on environmental contributions to ME/CFS and Long COVID. There are many misconceptions about what an environmental illness is, so I just ask everyone to keep an open mind and please provide me an opportunity to fully lay out the case for environmental considerations. And I hope to be able to change your perspective. If we were to consider ME/CFS and Long COVID as an environmental illness, what would that relationship be and what is the mechanism of how symptoms present? I believe the link here lies with environmental exposures mediating symptoms that individuals with acquired barrier damage become susceptible to. One of the most common phrases that you might hear when you ask a patient to describe their symptoms is it feels like there's toxins or cement in my veins. My question to everyone is, why are we so quick to dismiss that? I think that claim has clinical significance and we should consider the literal implications of it. Environmental exposures mediating the symptoms in ME/CFS would also help to explain the heterogeneity and diversity of symptoms in the disease. What factors a person is exposed to determines the symptom profile the patient will develop. Before we move on, I wanted to clear up prior misconceptions about what an environmental illness is not before I introduce what it actually is, and I'll provide evidence for these statements later on in the presentation. An environmental illness is not an allergic reaction. Inflammatory responses may occur, especially in the case of mast cell activation syndrome, but it's not a requirement for an environmental illness. It is not a somatic or sensitization disorder. If it were a sensitization problem, you would expect the same symptoms from different exposures, but different exposures result in entirely different symptoms. It's not an acute reaction. While you can have acute symptoms from exposures, symptoms of environmental illness persist chronically. This can either be from chronic exposure in the case of proximity to pollution or exposures in the home, or an acute exposure that results in chronic symptoms. Also, it is not a gimmick or spiritual healing. This is not getting rid of bad chakra in your house. This type of research needs to be taken seriously. What does an environmental illness look like? Many illnesses for which the root cause was previously unknown are starting to be understood by environmental factors that drive them. Take, for example, Alzheimer's disease. This figure is from a study on the brains of patients who had familial Alzheimer's disease. They looked at the location of aluminum, which is a neurotoxic metal that should not be found in the body at all, and amyloid beta plaques, which are protein aggregates that have been found to drive the progression of Alzheimer's disease. And as you can see, in every spot in the brain that those plaques were found, aluminum has been found as well. Alzheimer's disease does not present with any chemical sensitivities, allergies, or immunological reactions, but this environmental factor has been shown to drive the progression of the disease. Another study found that exposure to a common dry cleaning chemical called trichloroethylene causes a 500% increased risk of developing Parkinson's disease. Another study found that people who have been diagnosed with irritable bowel syndrome have 50% more microplastics in their gut than people who haven't. And more recently, exposure to a nerve agent called sarin gas has strongly been implicated in the development of Gulf War syndrome. We have many cognitive biases when it comes to human health, particularly from an environmental standpoint. Take, for example, when it comes to farming. If you find a plant that's sick, it's always presumed to be an environmental problem, whether from pests, drought, lack of sunlight. However, when a human gets sick, it's never considered to be an environmental problem. It's just presumed that anyone that gets ill just has a manifestation of genetic issues that accumulate over time sporadically. I think it's very important to acknowledge and try to remove these cognitive biases and strongly consider the case for environmental factors driving this wave of chronic illnesses. How can we characterize an environmental illness in the scope of the many different types of modern day illnesses that our population faces? I produced this model to help conceptualize it. In this model, what type of response our body has to a given exposure determines what the resulting disease will be. If you take a cell and expose it to a chemical, that cell then becomes damaged and the rest of the body loses the function that that cell had. If the body tries to compensate for this loss of function by hyperproliferating, the result is cancer. If the body tries to clear away contaminants or disease cells by using the immune system, the result is autoimmunity. If the body cannot compensate for the damage, the result is a loss of function and ME/CFS. This understanding is important because it would suggest to us that we might need to redefine what different illnesses are. If someone is exposed to the same environmental insult but develops two totally different sets of symptoms due to differences in genetics, are those different diseases? If somebody is exposed to two totally different insults, but the damage results in failure of the same processes, and the symptoms are the same, are those different diseases? With this understanding, I propose that ME/CFS is an illness caused by damage to the body rather than dysfunction, which is the typical assumption that researchers make. Irregularities that are seen in these patients are either a sign of loss of function from said damage or a compensatory attempt to replace that function, such in the case of hyperadrenergic neurological responses. For those of you who might be unfamiliar, I'd like to give a quick introduction on what an epithelial barrier is. Our skin cells are held together by proteins called junctions that play a vital role in protecting the body from foreign substances, including pathogens and pollutants from entering the body and making us sick. Disruption of these barrier junctions has already been implicated in allergic, metabolic, autoimmune, psychiatric diseases, and more. There are many factors that can damage this barrier, including but not limited to pathogen and viral infection, chemical exposure, and connective tissue diseases. If our body's barrier integrity is compromised, the contaminants will trigger activation of an enzyme called ACE2, which plays a role in SARS-CoV-2 entry to the cell, initiation of inflammation, and changes in blood pressure. So even local changes to a barrier will lead to systemic changes in the body. I created another illustration here to depict the role of the epithelial barrier in neutralizing VOCs, which stands for volatile organic compounds. They are chemicals that people can inhale and frequently expose to in their daily life. This illustration shows how small exposures may result in a progressive condition. These epithelial cells may be acting as a buffer against toxic and chemical exposures. When the barrier is damaged enough, the buffer is no longer sufficient to be able to neutralize low-level VOC exposures. And if the rate of inhalation is the same and the buffering capacity decreases, then the toxicity increases. Even in models where the barrier was initially damaged by an infection, it may have reduced the buffering capacity of the tissue enough that low-level VOC exposures that the body was previously able to neutralize then become harmful and pathologically relevant. This may also have enough of an effect to keep the tissue from regenerating after a damaging event. I mentioned earlier in my presentation that different symptoms correlate with different types of exposures. I'd like to cover the three largest triggers for symptoms based upon my personal experiences. The first one we've all heard about is mold. Mold will cause flu-like symptoms, including feeling feverish, lots of malaise, and what I can only describe as this vascular pain. Common sources of mold exposure include between wooden panels in the bathroom and air vents, just to name a few. Mold exposure can occur from mold both inside and outside of the home. And the second type is VOCs, which I previously introduced. They produce a very stimulated feeling when exposed and a feeling of chest compression or flattening and an inability to rest or recover. These chemicals cause the barrier damage that leads to susceptibility to other environmental toxins. Common sources include, but not limited to, varnish, memory foam, vinyl, fragrance, and carpeting. The last type I consider to be the most hazardous. They are particulates. They create a feeling of fullness or pressure in the body. They increase pain, fatigue, and weakness, and create a heavier, clogged feeling. I believe these to be the main culprit that triggers symptoms in ME/CFS. This category of exposures is unique because it is the only one in which symptoms persist or worsen even after you're removed from the source of exposure. Common sources include, but not limited to, painted glass and metal, renovations like sanding or brick cutting, and especially matte white paint. Because particulate exposure is the least known offender of the three I listed, I wanted to explain exactly what it is. To use just as one example of a particulate exposure, I'd like to bring your attention to titanium dioxide. Titanium dioxide is an inorganic powder that's chemically inert and also very hard and abrasive. It's one of the most commonly used particulate compounds found in many consumer products, including cosmetics, food, toothpaste, sunscreen, paper, soap, shampoos, tattoos, and paint. This substance was banned from the use in food in Europe because of its known risk of causing cancer, but we still frequently use it here in the United States. Studies have shown that it is not absorbed well when ingested, at least in healthy subjects, but it is rapidly absorbed when inhaled. As a result, I believe the biggest risk of exposure then comes from matte white paint, which can release particulates from surfaces for years, which are small enough in size that they will stay airborne indefinitely and have no smell. I want to take just a second and ask everyone to think about, what do you think would happen to you if these nano-sized, chemically inert, abrasive particulates were to get inside your body? I think that the answer is that it would look a lot like ME/CFS, and the damage it would do would be by mechanical means, not reactive ones, so the effects would not show up on standard blood tests. Here are some clinical signs I've personally seen in myself that I feel are supportive of the mechanism I've outlined. First, in order to establish an environmental illness as a biological entity and not a psychological problem. This is a photo I took of my arm after I left a standard bandage on it for a day. The area of skin that was in contact with the adhesive from the bandage started to fall off without any signs of inflammatory or allergic response. This makes it a biological toxic susceptibility to chemicals that was acquired. I've used a million bandages in my life and I never had a problem with them before developing ME/CFS. This chest CT scan I had shows the area between the trachea and the sternum is significantly smaller and the thymus is compressed relative to healthy control images. I believe this is medically relevant, and this chest compression is a result of the tissue in that area being depleted from damage caused by chemical exposures. This compression of the chest could also impair the thymus, which helps to develop the immune system, as well as compress the internal mammary artery, which would reduce blood supply to the chest, further exacerbating the progression of the disease and contributing to shortness of breath symptoms. Repeated brain MRIs show flare hyperintense foci in the white matter, which are a non-specific finding, but one that is very common to ME/CFS patients. I came across this paper that says that these white matter lesions are a mechanical loading state of the ventricular wall that causes ependymal cells to be stretched thin and is strongly indicative of a mechanical contribution to ventricular wall failure. This means that those hyperintensities are areas that were mechanically stretched thin, and I believe this is due to the buildup and clogging of particulates, such as titanium dioxide. For reference, my own personal health condition worsened substantially when I moved into a new apartment. When I went there, I was fine in that environment. However, the landlord sent in some painters and painted all the walls with matte white paint. And my condition became quickly very substantially worse. And it would worsen further every time I took a breath in that environment. This exposure event was like an experiment, and all of the variables were controlled except for the use of matte white paint, which is heavy in titanium dioxide and contains a relatively low ratio of binder to pigment. Upon moving into that environment, I noticed these red dots all along my arms that were only visible for a few minutes after I woke up. They were not itchy or elevated and appeared to be small spots of bleeding underneath the skin. I then came across a study that shows that titanium dioxide exposure causes endothelial cell leakiness by disrupting cellular junctions. I also noticed I developed these extreme striations in my skin that I didn't have before. They were worse shortly after the exposure event, but have faded mildly since that time. Another study showed that titanium dioxide exposure causes separation of muscle fibers, and the pattern of muscle fiber separation appeared to match the pattern of striations on my hands. Another abnormality that I've seen in many other ME/CFS patients are chronically low organic acids, which are metabolites from energy production. I found this study that looked at metabolites in rats that were exposed to titanium dioxide, and they have a similar profile. Within days, their organic acid levels depressed and persisted low indefinitely. I then took a look at where titanium dioxide ends up in the body. This study found that it will concentrate in lymph nodes far more than any other organ or structure. And I thought this was interesting because I remember reading some papers published by an osteopathic doctor in the UK who found numerous clinical signs of lymphatic dysfunction in his patients for decades. He claimed that lymphatic dysfunction is visible in approximately 80% of ME/CFS patients, and even included this photo of one of his patients that shows engorged lymph nodes, and so it should be considered a strong candidate for causing this clinically observed lymphatic dysfunction. To summarize, I think that ME/CFS has a two-stage mechanism to develop the condition. The first is barrier damage, which increases the person's susceptibility to contaminants in the air. And the second is exposure to particulates, which cannot enter the body without barrier problems. When I had moved into that painted apartment, my partner was completely fine while I significantly worsened every day, meaning I had to have a particular susceptibility to an environment that my partner did not have. And I think that previously acquired barrier damage was that susceptibility. When these particulates enter the bloodstream, they will wedge in between endothelial cells, promoting blood vessel leakage. I made a short video that shows nanoparticles moving through the capillaries where they diffuse into interstitial spaces between tissue. Some will get lodged here and some will be drawn into lymphatic vessels where they can be clogged in the gated lymph nodes. I like to think of it as snow in a snow globe. If you shake them up, they can travel throughout the body, but by default they are not bioaccessible and tend to remain lodged in these spaces. The result is poor perfusion because those sites are physically inaccessible for oxygen delivery. These particles may also perforate connective tissue, leading to ligament laxity, compression of nerves and blood vessels, and even displace epithelial cells so they cannot form junctions properly. The mechanism I'm outlining doesn't have to be at odds with other modern mechanistic theories in ME/CFS. There may be a little bit of truth in most or all of them. Here's an example of how I think they might relate. Most of us are probably familiar with the cell trend adrenergic autoantibody test for ME/CFS. It's been around for quite a few years now. Well, a 2021 paper implicates cell trend adrenergic autoantibodies as markers of barrier permeability, citing gut bacterial antigens, which are structurally similar to human adrenergic receptors, may leak through the intestinal barrier and lead to the production of autoantibodies. Many of us are also familiar with the metabolic trap theory proposed by Ron Davis. His model proposes that in bimodal metabolic processes, an initiating factor can shunt one pathway, leading to a perpetual cycle in the latter. He has had a specific focus on the kynurenine pathway as one potential culprit. Now, the paper I showed also proposes that gut barrier dysfunction would result in increased tryptophan absorption, which would be the factor that increases substrate levels to trigger the shunting event in Ron's metabolic trap model. If his model of the overreaction of the kynurenine pathway is true, then it really could just be another marker of barrier permeability. I wanted to share some imaging that I personally did, which produced some really interesting results after nearly 5 years of work. I conducted TEM Imaging, which is a type of electron microscopy, of a 3mm punch biopsy of skin that I provided myself. In the images in A, there is an abnormal mass accumulated in the cytoplasm of a Schwann cell coating a myelinated nerve. I ran these images by about a dozen top nerve morphologists, and they all agree that accumulation is abnormal. It's obvious from the photos that the mass has become so large, it's actually pressing on the nerve. The black dots in the mass consist of glycogen deposits, which are a form of energy storage. Glycogen deposits are sometimes a feature of degenerative axonal neuropathies, and being a source of cellular energy would fit into the picture well of ME/CFS being an energy production problem, although they may just be a consequence of poor blood flow or oxygenation. It's unknown what the white clumps are, but I think they're most likely Nissl substances, which are a consequence of protein folding problems. If there's actually a problem with protein folding, however, these cellular problems likely go beyond energy production and potentially may be a sign of toxin exposure. In the images in B, you can see some large pockets in the myelin. They were seen both in the stained sample on the left and the unstained on the right. Now, these pockets may or may not be abnormal. The peripheral nervous system does have a feature called Schmidt-Lantermann incisures, which are very stable openings within the myelin. However, in my personal opinion, the openings do not look stable or organized. They also do not look like typical demyelination seen in MS or dense degeneration. I personally think they could be a sign of mechanical damage, either a puncture or tearing of these nerve fibers. In image C, there's also a keratinocyte that displays a very large accumulation of glycogen deposits, which is so large it virtually takes over the entire cytoplasm. This is very relevant to the mechanism I've outlined today because keratinocytes play a critical role in the formation of the epithelial barrier. Lastly, in D, I just wanted to include an image from the unstained sample shown on the right that contains a group of dots free in the extracellular matrix, which looks strikingly similar to titanium dioxide nanoparticles as seen by TEM shown on the left. I cannot conclusively say that's what they are from this image, but it is suggestive and I believe warrants further investigation. The existence of amyloid fibrin microclots has recently been linked to CFS and Long COVID, which potentially clog capillaries and deprive tissue of oxygen. You can see images of the microclots on the left from a commercial test. So to investigate this further, I purchased my own microscope that I could take some nail fold capilloscopy images to directly observe the effects on the capillaries. And I thought it would be cool just to show a quick clip I recorded myself of red blood cells moving throughout my capillaries. And it may be a little bit difficult to see over a Zoom call, but they're moving extraordinarily slow. I continued to regularly monitor my nail fold capillaries, checking them every day for about 10 months. And what I noticed is when I went through these periods where my symptoms got substantially worse, I'd find these black dots inside of microhemorrhages. It's possible they are hemosiderin deposits, which is an iron-binding protein, but they're very close in size to the dimensions of the microclots that were measured in the commercial assay. So I do wonder if what we're seeing here are microclots themselves. This got me thinking about what role microclots could play in the mechanism of ME/CFS that I proposed. I did my homework and came across a study that said we have found both albumin and fibrinogen interact irreversibly with the negative surface of titanium dioxide nanoparticles. So I wonder if the titanium dioxide nanoparticles are actually what is nucleating the microclots. They wouldn't be able to break down when they're irreversibly bound to the fibrinogen, but they also wouldn't go on to form regular clots because there's no clotting factors activated to expand them. Now I have to address the concern that I know many people that are thinking when they hear a pitch on environmental drivers, which is now, wait, I don't have any of these environmental sensitivities. I got sick from a virus. My problem is viral persistence. So here's how environmental factors can still play a role in an illness that was initiated by a virus or other infectious event. Using titanium dioxide as an example, titanium dioxide exposure increases the susceptibility of epithelial cells to bacteria and viral infections. Even short-term exposures to particulate matter at a young age increases the risk of infections, and co-exposure of cells to titanium dioxide and a virus simultaneously causes more disruption of the junction integrity and increased viral infection compared with either treatment alone. All of this means that environmental exposures can increase the risk of infections, and the damage that infections and environmental exposures both do to the body is additive. Looking at particulate matter more generally, pollution induces oxidative stress, impairs epithelial barrier function, reduces antiviral responses, induces immune suppression and inflammation resulting in more severe viral disease, and also suggests theoretical implications, including the possibility that particulate matter may carry SARS-CoV-2 deeper into the lungs, and could induce DNA damage in the virus, altering its genome and increasing infectivity. Another paper notes that the virus can overcome the alveolar barrier in severe COVID disease courses, and neighboring endothelial cells are affected by epithelial cytokines, which are signaling molecules, which further results in the damage of epithelial and endothelial barrier, which, remind you, is the feature that causes future environmental susceptibilities and viral dissemination. So there are many ways that environmental factors can play a role in a viral-initiated illness. So what is the relevance that my personal observations and research has to the patient population as a whole? I ran polls in 2 different ME/CFS groups online, and 80% of the ME/CFS community consistently self-reported as having environmental sensitivities, with 178 people voting in total. Because it's likely difficult to identify the source of symptoms that you're exposed to chronically, it's potentially with many different triggers or something you've previously been exposed to. It's possible the actual number could be 100%. So with nearly 100% of patients affected, why is nobody else studying the subject? I traveled up to UMass earlier this year and spoke with a brilliant researcher named Dr. Anna Gill, who is running the Inspiritol trial for ME/CFS. When I spoke to her about the subject, she said, "I've never come across an ME/CFS patient who didn't have sensitivities of some kind." Environmental illness is often not easy to discern. I didn't notice any environmental sensitivities for the first 4 years of my illness. Another person here said it took me over 20 years to realize that my ailments are directly related to the external environment, to the air I breathe and to the food I eat. I spent the next 20 years trying to figure out exactly what was causing those reactions. Looking at some other testimonies, one person says, "If I were living in a less congested area, I think I would be better off. As it's been said, avoidance is the cure." Another says, "Long COVID here, environment does seem to make a huge difference to me. Every year I've camped in the forest for a couple of weeks near the sea, and by midweek my symptoms have improved." And another says, "While my issues started from COVID, it seemed to make me much more susceptible to triggers in the environment." I've heard of many similar stories from patients with all different diagnoses, and I've spent a substantial amount of time in long COVID group chats, and I've heard many stories from even people who are big believers in viral persistence about worsening in their health condition following exposure to cleaning chemicals and the like. I'd also like to play for you a short clip from a video interview I had with a person who has had long COVID for over 4 years. I was friends with him for nearly 2 years of his journey, getting weekly updates on how his health was deteriorating. He has never had multiple chemical sensitivity nor any diagnosis besides long COVID, and he didn't have any known environmental sensitivities, including during the period that he had long COVID up until the date of his move.
I starting understanding and believing Scott in a way that I should definitely try this. It's not gonna hurt. Try a way that it's healthy and it's gonna help you probably. So yeah, after we had multiple conversation, I decided to go to a more better quality air, and when I went there, it didn't even take that long, like 3 to 4 days, and I started feeling that fresh of air I was breathing. Like I went to more of a sea place, like where it's like the ocean, and there was no cars moving or so yeah, like in 3 4 days I was like starting breathing better. My brain fog was getting way more better, and I also eliminated some sugar and those other stuff that Scott mentioned. And yeah, it really changed me. Honestly, when I heard the message that you read before and after, hearing that, I don't see that person anymore. I don't see. I believe it. I believe him because it is true, but I don't see him like. I'm glad I don't see him in that spot anymore.
As you can see, upon moving to better air quality and away from pollution, he has entered remission and is still in remission today. I wanted to cover what the treatment implications would be for people with an environmental illness, which would be a radical shift from current medical practices. There would have to be a treatment shift from putting toxins into the body, like pharmaceuticals, to removing them. Doctors need to advise patients to check their homes for environmental contaminants and switch to safer products or housing when possible. But in our current medical system, what doctor would ever think to advise that? There would be a few points of resistance to making that happen, including from pharmaceutical companies, as avoidance isn't profitable, and even from patients, as psychologically it may be easier to scapegoat a virus or swallow a pill. However, this information would also bring power to the patient, as they would have some degree of control over their own health. Avoidance is not, it also won't have any side effects. Not being exposed to toxic chemicals, it never hurt anybody. Housing would need to be covered under health insurance as a medical device, not just shelter. And finally, there would need to be proper regulation of building materials and consumer products, for which there is almost none. You might think that when you go to a store and buy a cleaning chemical, for example, there's some entity overseeing its safety, but none exist. Thousands of toxic chemicals are hidden under the label of fragrance or proprietary blend, or can be in the product as an unintentional byproduct of manufacturing, which means these companies don't have to disclose any of those components. These pictures are taken from the very first 2 bottles of cleaning chemicals I picked up at my parents' house that contain ingredients that manufacturing companies don't even disclose. However, many companies don't even care this much. Some will just put 98% other ingredients, and that's all they feel like doing. I was exposed to this product about 2 weeks ago while staying at a hotel. The owner there said there's no chemicals in this bottle, it's just deodorizer. Because of how things are marketed, labeled, and hidden, the average consumer doesn't even realize they're being exposed to toxic pesticides, among other unknown ingredients. Obviously, these companies will strongly oppose any increased regulation, which will need to be substantial. There also needs to be a reassessment of what we consider to be safe. Whether or not a rat develops cancer and dies in 30 days is not a sufficient assessment of safety. We sit and breathe in many of these chemicals on a daily basis for many decades, which can have long-term consequences on health outside of acute toxicities. Additionally, we're exposed to many different types of chemicals and pollutants simultaneously, which could cause additive damage. These studies also need to include consideration of individuals who may have genetic variations that predispose them to be susceptible to particular exposures. Avoidance is not the only thing we can do, but it's a place to start, and anyone can do it without tons of money. In the future, we can develop therapies to filter the blood, sequester metals, unclog lymph nodes, etc. But we can't even get to that point if we're not funding this area of research at all. Thank you all very much for taking the time to listen to me today, and I hope you've learned something new, and the research and information I've presented here can be relayed to other researchers that can study this area of environmental medicine properly to help the lives of patients who need help today. Thank you very much for your time, and if anybody has any questions, feel free to comment or send me a message.